This set of slides provides an introduction to the nature and theory of connectrons. The genesis of the idea was about 13 years ago when I was looking into the molecular biology of the cell and thinking about a variety of things. I developed a sequence-based method for finding RNA and DNA patterns that could decide which genes are open to expression. I looked in the molecular biology of the cell, and there was this beautiful diagram of the 6 levels of the structuring of DNA, all of which are familiar to you. But at level 5, they said, at level 4, they said these loops are random in length. And I said to myself, oh, no, 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 they're not random in length. You just don't know why they're particular lengths. And so I set out to try to show that was true. This is the model that the molecular biology of the cell shows. It's 30 nanometer loops of chromatin structure, and then a protein structure that holds them together. But nowhere in the molecular biology of the cell did they tell us why these loops were a particular length, and so that was the investigation that I performed. And they also said that sometimes these loops open up like this, and so I thought about why that was true. There could be other constraints, not at this level, but up in here, which were also acting to control expression. So this is the molecular biology of the cells model. So what's the role of connectrons? Well, promoters and their associated transcription factors regulate the quantity of a gene that gets expressed. There's promoters that have a little bit, that make a little bit of the following transcript, and some that make a medium amount, and others that make a lot. So it's basically an analog process. RNA interference is mostly a post-transcriptional mechanism for regulating the amount of RNA that gets translated into protein. So my argument is that connectrons are another mechanism, more closely related perhaps to cellular differentiation. So I've been designing zinc-finger DNA binding proteins and gotten a patent for that, and so I try to think about how these zinc fingers could be applied to closing these loops. The trouble is, though, that the couple-like DNA binding proteins, there aren't very many of them. And they're expressed at different levels of cellular differentiation, but there just don't seem to be enough of them. And so I did some computational experimentation to show that there are a finite number of sources of RNA and then pairs of DNA target sites, which together could make an effective gene expression control mechanism. First in yeast, and then later in many other genomes. I've shown that these level 4 loops range out to several megabases from about a half a kilobase. And specific double-stranded sequences exist on the left and right flanks of each level 4 loop. And that an RNA is generated either by gene coding or non-coding DNAs that form a pair of triple-stranded Hook-Steen helices that stabilize the 30 nanometer chromatin structure. So this is the idea. You can have a gene coding or a non-coding element, and then 2 signals, C1 and C2, control 1 and control 2, which are normally adjacent to each other but can have mutational gaps in them. And these transcribing elements can be anywhere between half a kilobase and 3 megabases in length. And then on the same chromosome, a different chromosome, you have 2 distinct sequences, T1, target 1, and target 2 on the right. And then in between you can have any collection of gene coding elements or non-coding elements or nothing. And these are the things that are going to form the loop. So the DNA gets transcribed into RNA. It can be from a non-coding source, or could be the gene itself, or an mRNA, or an intron. And it turns out normally the introns are the sources of the connectrons in the higher eukaryotes. And so what happens then is that the RNA, 2 signals, C1 and C2, move around in the nucleus, close perhaps to where they were generated, and find a cognate DNA double-strand sequence. So the C1 sequence and the T1 sequence are the same sequence in the direction of the major groove. And then the C2, if the T2 is close to T1, then the C2 can wrap around T2, again in the major groove, and form another triple-stranded Hooke-Steen helix. And so this set of 4 sequences is a connectron. And then the loop formed by this structure is in 30 nanometer chromatin structure, and it can condense down and even be pasted down by methylation for permanent closure if the system wants that. So this is the complete theoretical model of the connectron formation process. And so I've gone about finding these patterns, finding if these patterns exist, and they do. Now, one of the things that molecular biology also talks about is the condensin dimer, which has the effect, as you see in the following here, to help to stabilize these loops once they formed. But the specificity of the loop length, that has to do with I think the connectron, because the connectron is lying down in here. And so this is the proteinaceous backbone that the molecular biology of the cell talks about. So as I said before, I mean, the methylation developments are consistent with the connectron hypothesis because they're concerned with pasting shut these loops of 30 nanometer structure. And whole regions of the genome can be silenced by this means. So this just shows how different portions of chromosomes can come or be brought together, and how they can function together because they are close, and how the behavior of the transcription occurs when pieces of different chromosomes are close to each other in nuclear space. Well, the connectron determination calculations on many different genomes and genome types show that connectron sequence tetrads exist in good numbers and in interesting places and in interesting relationships. Connectrons provide a programming language for the temporal control of gene coding and non-coding expression in cells.